Thank you so much for the invitation to this meeting. Thank you, Esper. This has been amazing. So again, as Hendrik said, I have some translations from Google Translate, and I hope they're correct. And again, thank you for Mike. I think this talk nicely segues, and I will spend almost no time on the background. I just wanted to make the point that there's this history of using monoclonal antibodies for hundreds of years for prevention or treatment. And so here's a slide on the history, and here's a slide to the same table that Mike showed, that monoclonal antibodies are being used and are being developed for many infectious diseases currently. So I'll quickly move to the virus we're talking about, which is HIV. And so a main question is, do we need monoclonal antibodies against HIV for treatment? Are they necessary? Many people say no, but I would argue that having an alternative form of therapy that could complement antiretrovirals is important and could be useful in certain cases. And some obvious arguments would be antibodies might be useful in mother-to-child transmission. They could be useful as a treatment break from antiretroviral therapy for people who have trouble with side effects or adherence with antiretrovirals. Antibodies can be optimized to have long half-lives. And actually take that a step further, antibodies can be optimized for lots of different functions that I think we're only beginning to explore. And finally, they have a different mechanism of action from antiretrovirals, and there's the possibility of not only binding to and neutralizing virus, but there's the possibility that they can bind and clear infected cells, and this is really an active area of exploration in the field right now. All of which leads to the question of, once you want to explore the use of antibodies, which antibodies to use? So first of all, a little background. Are all antibodies against HIV equal? And the answer is no. And so this is a summary from many, many studies by many groups who have really in detail explored the antibody response to HIV infection. And in general, you can find that people are infected with very few numbers of viruses, often one, founder virus. And the antibody response is usually very specific against this one founder virus. As the antibody response grows, the virus easily mutates, and so you begin to generate diversity within a person. And so here you can see the virus is starting to mutate away from the green virus. I'm just using colors here to represent the quasi-species. Virus mutates away from the green antibodies, and then you start to develop a blue antibody response, and so on, until, in general, when you look at someone's antibody response, most of the antibodies are very strain-specific, meaning the green antibodies neutralize the green virus, the blue antibodies neutralize the blue virus, and so on. And so how do we know this? Oh, sorry, and I just want to make the point, so this is really a coevolution constantly between virus and antibody. How do we know that these antibodies are neutralizing only certain viruses? Just a little background on the assays that we use, or really one assay, the neutralization assay. The general concept is that we have two plasmids. One plasmid, it contains a genome of HIV that is OMV deficient, and another plasmid contains the OMV gene of interest. When we co-transfect these plasmids, what we get is a pseudotyped virus that express the OMV proteins of interest, and they're only capable of infecting one time. And what we do is incubate these viruses with target cells that express high levels of receptor, CD4 and CCR5, and these cells, upon productive infection, will express luciferase. And so then what we can do is titer in monoclonal antibodies or serum and measure how much luciferase expression is decreased and generate these neutralization curves. And then you can see that as the curves shift to the left or right, we can also learn about how well antibodies neutralize virus. And we can summarize this by taking 50% or 80% neutralization. What is the concentration of antibody needed to neutralize that much virus? And so when we start to talk about breadth and the ability of antibodies to neutralize lots of different viruses and take into account genetic diversity, Here's just a schematic showing some of these large virus panels that have been developed. Here's a dendrogram showing the genetic diversity on a panel of 174 OMV genes from all different subtypes. And so you can take antibodies and test them in this system against all these different genetic variants. And so, again, this is just one summary figure from work that's been shown by numerous groups, and that is that there's a continuum of responses against HIV when you look at people who are infected. And so here is just a figure from the Los Alamos group who took 117 serum from chronic infection, looked at 219 genetically diverse viruses, pseudoviruses, and then here what they're graphing is the percent of serum that neutralizes this many percent of viruses. And so one important point is that 100% of serum neutralize something. Almost every person makes an anti-HIV response that can be detected against very sensitive viruses. But then there becomes this continuum, and some of the numbers that are thrown out, I just want to highlight 50%, this dotted line. About 50% of people can neutralize about 50% of genetically diverse viruses. So there's about 50% cross-reactivity. And then when we start to talk about breath, somewhat arbitrarily, we get down here, and the numbers that are thrown out are about 20% of people here, 20% of serum, can have about 80% to 100% cross-reactivity. And so this is the area where we define as people who have breath. So that's where that comes from. And so next, now that you can identify these people, can you pull out the antibodies? And again, this is just a schematic of methods where basically people have looked or pulled out either epitope-specific B cells or just B cells in general. You take B cells from the blood, isolate RNA, generate cDNA libraries, and then amplify heavy chain and light chain genes of these B cells, clone them into expression vectors, co-transfect the heavy chain and light chain, and then you have a monoclonal antibody, which you can then go back and test in a neutralization assay and show that it's broadly neutralizing. And so a lot of groups have done this. There are many, many broadly neutralizing monoclonal antibodies that have been isolated. Are all broadly neutralizing antibodies against HIV equal or the same? And the answer, again, is no. So again, this is just a different version of a slide that Mike showed, where I'm going to show that most of the antibodies that have been identified have also been structurally characterized. And you can see that they neutralize all different forms of, all different parts of the virus. And so here's the virus membrane, and here's the virus spike sticking out. And we have antibodies against the GP41 membrane proximal region, antibodies that are called interface antibodies. They bind both GP120 and GP41, antibodies against the CD4 receptor binding site, antibodies that target the very top of the virus, V1, V2 apex, and antibodies that target what's called the V3 glycan. So again, you can see that the virus spike is covered in these sugars in green, glycans, and that some of the epitopes involve glycans. So it's both virus protein and glycan. And so now if you go back and test these new generation of monoclonal antibodies in this in vitro assay, what you can do is basically draw a graph of at each concentration of antibody, how many viruses they neutralize. And then sort of, this is a way of distinguishing between different broadly neutralizing antibodies. And so I just want to make a couple points that sort of previous antibodies that we had were not so broad. If you just look at the very end of the spectrum, at the highest concentration, 50 micrograms per mil of antibody, the breadth of, say, B12 or 2G12 is not very high. And then you can see the difference here, really, between some of these new antibodies that neutralize very, very many viruses. But another really important point for moving into the clinic is that as you move down in concentration, the potency of these antibodies differs. And so I just want to point out here, in particular, PGT121. If you just look here, you'd say, oh, maybe it's not that broad. It's kind of in the middle. It only neutralizes some viruses. But as you move and follow this line and look down here, it takes very little amounts of this antibody to neutralize about 20 to 30% of viruses here. And it's much more potent than other antibodies. And so these factors that we see in vitro may be important to think about as we move to the clinic. And then again, another point is that was just a summary of a large panel of genetically diverse viruses, but you can also break that down by subtype and see many differences in these antibodies. And so here's just a table I made of a couple different antibodies that are thinking about going into the clinic, and if you break up that previous data by subtype. So one good example here is VRC2625, which targets the top apex of the virus. And if you look at, say, subtype B, percent of viruses, it neutralizes by 80% with less than 10 micrograms per mil, is very low in subtype B, 13%. But if you look at subtype C viruses, it's much broader at 65% with incredible potency. Whereas you look at some other antibodies, and here I'll just want to point out 3BNC117 and VRC01 are two that have been moved into the clinic and tested against many people in North America, so mostly subtype B infected. And what you can see is to neutralize in vitro 80% with less than 10 micrograms per mil. They're both pretty broad, about 90% breadth, but there's some difference in potency, and 3BNC has a geometric mean IC80 of 0.2, while VRC01 has a geometric mean IC80 of 0.8. And again, this may make a difference. And so then just briefly, studies of using monoclonal antibodies to treat chronically infected people have been done before. And again, it's been done by a couple different groups and many different papers, and I just want to highlight one study and a couple different factors when you look at these different studies is how many antibodies. This was a cocktail of 3 antibodies, 2F5, 4E10, and 2G12. There were multiple infusions, and these people were pre-screened that they had some sensitivity in vitro to these antibodies. Here's just a summary slide of what they found. This was a study in analytic treatment interruption. So they took people, and this graph here in particular is for acutely infected individuals who went on antiretrovirals. They were given multiple infusions of the antibodies, and then the question was, did the antibody treatment delay virus rebound or decrease virus load? And they saw a delay in rebound, and here are the two control group versus passive immunization. And then they also could detect that virus escaped from antibodies and became more resistant. So question after all these groups have isolated these new broadly neutralizing antibodies is, can we repeat some of these studies and do we get different results? And here are 3 papers that have been published in the past 2 years, all with a single infusion of an antibody. So it's 3 different antibodies, 3BNC117, VRC01, and 101074. And these are all in people who are not currently on antiretrovirals, so they have replicating detectable plasmaviremia. And so I'll just go through a little bit of this data, but I'll focus in my talk on the VRC01 studies because those are the samples I analyzed myself. Here's a summary slide from all 3 different trials. And what I'd really like to point out is that for the most part, you see the same thing. And so I'll start with VRC01. So here is, from baseline virus load, what is the change in virus load over time after you give someone an antibody? And it's about a log. And so the black line here is the average of everyone in this trial. You look at data from the Rockefeller group with 3BNC117. They did these trials with 2 different doses of antibodies. So this is a lower dose of antibody and a higher dose of antibody. And they're showing the average in red. And what you can see, again, about the same is it's about a log decrease for everyone. They just published on 101074, so it's a different antibody. They, again, did 2 different doses. Again, in red is the average, and again, it's about a log-fold drop before virus rebounds. And so I'll just point out there are a couple differences in these studies. These people were pre-screened for sensitivity. Here, these people were not it's my understanding these people were not pre-screened, but for data analysis, they took out people who were resistant. And here, we just lumped everyone in, so it's including these 2 people who were resistant. But even taking into slight differences in how these trials were run, the overall effect is exactly the same. So one question is, when we talk about responders and non-responders there and pre-existing screening, does it matter? And the answer here is yes. And so I'll just show some data from the VRC01 trial, but this is applicable to all 3 trials. So the data I'm showing is from the NIH trial at the VRC, where we had 8 viremic donors, singly infused 1 time intravenously with a high dose of antibody. And then what we did was isolate OMV genes from plasma from before and after infusion with the antibody. We then could look at sequences, so we generated single sequences, and then we took some of these sequences and cloned them and expressed the OMV genes in the pseudovirus assay and looked at sensitivity to antibodies. And so just quickly to show that everyone was subtype B infected. So here's just a dendrogram with alignment of heterologous subtype B OMV genes in gray. Each of the participants are in color, and they have a large diversity to start with that is consistent with being chronically infected with HIV. So this is the genetic diversity within each subject. Then when you take these OMV genes and clone them and look at sensitivity to VRC01, I'll just highlight 2 points that indeed having a very high IC80, i.e. being pretty resistant in the in vitro assay, these were the 2 subjects who had the least virus load decrease. So there was an absolute relationship between being resistant in vitro to having almost no drop in viral load in vivo. But then when you look at these tiny differences with people who did respond, there was no correlation between small differences in IC80 and slope of virus load decline or magnitude of virus load decline, and whether that's small numbers or it's sort of a yes-no threshold is a question that we still don't know. Oops. And so the question that I really want to focus on, that I'm very interested in, is how does the virus replicate in the presence of antibody, i.e. how does it escape? And so here, I briefly want to show actually the latest data from the Rockefeller group with 101074. This is an antibody that targets the V3 glycan, and the epitope is it's been very well documented in vitro, and the epitope is fairly simple compared to the epitopes of some other antibodies. And so here, what I'm showing is a figure from that paper where they look at the epitope of the antibody, and really they're actually only looking at 3 residues, 325, 332, and 334, over time in these people, and then the percent of mutant versus wild type is shown, where wild type's in gray and mutant is in color. And so each residue, so changes in 325 are shown in red, 332 are shown in blue, and 334 are shown in green. And the concentration of the antibody is here in white. And so what I want to highlight from this figure is that almost everybody starts with a 100% wild type sequence that is sensitive to this antibody. Immediately, you infuse people with antibody, and the virus generates an escape mutation. One thing to note is it's mostly green and blue, so really all the escape is occurring in 332 or 334, which is the glycan. So essentially, the glycan is part of the epitope of this antibody. All the virus needs to do is change the glycan. It requires a single amino acid mutation. Every single person does it, and that's all that's needed for the virus to escape. And then another thing to note is just as antibody levels decline, and many people wild-type sequences come back, so it's very well highlighted in this person that it changes back to gray, but in this person it doesn't, the mutation stays. And so in all these studies, the virus usually always returns to set point as the antibody decreases, and there's a question of, is there a fitness cost or not? And it's sort of hard to say, because some people, it doesn't seem to matter that they have this mutation, and some people it does. Okay, so all that being said, is I want to explain why I'm more interested in antibodies that target the receptor of the virus, because it is a functionally conserved area of the virus, and the epitope's a little more complex. So here's a slide showing that there have been these class of antibodies called VRC01 class of antibodies, and what they are is all antibodies that target the same receptor binding site, and they're very genetically similar. But these are 9 different antibodies that have been isolated by different groups from 5 different people. And so it's very striking how similar these antibodies are genetically and structurally. And so to highlight the similarity, Tongjing Zhu from Peter Kwang's lab is overlapping all these structures on top of each other. This is 9 different antibodies overlaid on top of each other, all showing binding to GP120. And it's very, very similar how they contact the virus. And it's mainly in loop D, loop V5, and CD4 binding loop. Those are the virus contacts. And so a question that I've always had is if the antibody targets this conserved region, are there limited pathways of escape for the virus? And to, again, visually highlight this point, if you look at GP120 here in gray, the outline of the CD4 binding site where it needs to bind to the receptor is in yellow. If you then overlay the epitopes of all these antibodies in red, and then color code them according to how broad they are, the point of this figure is that the antibodies that are the most broad and the deepest red really completely overlap with the CD4 binding site, which I would hypothesize would make it hard for the virus to generate mutations in this site. So here, just briefly, I'm going to go through this data because it's already published. But these are two people who sort of fit that hypothesis, where in the VRC01 trial, they start off with low baseline virus loads. They're infused with, virus load is graphed on the left y-axis and is shown in color. An antibody concentration of VRC01 is graphed on the right axis and shown in black lines. And then sensitivity of the virus is shown in these gray bands. And so what you can see with these two people is that as antibody concentration declines, virus load returns. So these people become completely suppressed for about 20 days, and then as antibody declines, virus returns. So I hypothesize this is a case where these people can't escape from the antibody or don't escape from the antibody. And then just to highlight, they're infused with a large dose of antibody that's much higher than their in vitro sensitivity. So they start off with sensitive virus. However, the majority of participants in this trial, that's not what happened. You infuse them with antibody, their virus loads all decrease, and then by day 9, their virus loads start to increase again in the presence of antibody, even though these people start off with sensitive virus and they have a large dose of antibody infused into them. And so again, if we clone the envelope genes from 1 month post-infusion, so the virus that's rebounding virus, what we see is in vitro, they all become more resistant. Almost everyone except this person, and there's a definite trend, unsurprisingly. And then we have the sequences of these viruses. We can look and see what are the changes, how is the virus doing this? And so here I'm showing just in 4 people, you can look 1 month pre and post infusion, what are the differences? And most of them occur within the epitope of VRC01. And so the ones that are actually in the epitope I've highlighted in gray, but most of these changes are near the epitope. And so I'll just go through in detail a couple of these patients. So here is someone who escapes in the presence of antibody. The viruses are becoming more resistant to VRC01. But an important point that I would like to make is that we then also tested a subset of these OMPs for sensitivity to their own serum, and they're still resistant. So the fitness pressure on this virus right now is not only from VRC01, but it's also from their own antibodies. So we can look longitudinally over time at the phylogenetics of this virus, and so basically pre-infusion sequences are open dots, and 1 month post-infusion are blue circles, and you can see they're sort of clustering differently. We can look within the epitope and see that there's a big change at residue 429 in the epitope, and that also there's a selection for shorter V5 lengths here. And then we can take the length of V5 and correlate it to our in vitro assay and see that shorter V5 lengths significantly correlated with being more resistant to VRC01. And then we can look at 2 months out at this time when VRC01 has left the system, and what we can see now is that in green diamonds, viruses are all over the place, and so there didn't seem to be much of a fitness cost. By 2 months, we see the V5, selection of V5 lengths come back. So just quickly, there's this other person who actually, at the time of baseline, was co-infected with 2 different strains, and so bootstraps here of 100% are shown by this asterisk, and what we see is that predominantly day 2 and baseline, all sequences are on this branch, and there's 1 baseline sequence here. By day 7 in turquoise, we see this branch starting to expand, and then by 1 month post-infusion, the entire majority population of virus is completely switched from this branch to this branch. And then, unsurprisingly perhaps, we see that these viruses become more resistant to VRC01, and they maintain some level of resistance. They're not completely resistant to their own serum, but they're pretty resistant. And interestingly, the one that's very sensitive to serum, this was the original baseline minority strain, this sequence here. So it really seems like there's an antibody response against this branch, so this is the minority strain. Then you put in VRC01, the fitness environment becomes different, and then this branch is favored and outgrows. But it does maintain some resistance to autologous antibody. It cannot maintain this level of sensitivity to autologous antibody. And then if we look at 2 months post, what we see is a flip where this branch again becomes the most predominant branch once VRC01 has left the plasma. And then just briefly to point out that even people who start off with basically resistant to VRC01, very little virus load drop, we still see selection pressure. And so these are the envelope sensitivities pre-infusion, and this is post-infusion. So the VRC01 is still selecting and changing the genetic diversity of the virus here. And again, this person is maintaining complete resistance to their own antibody response. And what we see here is this is the diversity of the virus pre and post-infusion, and we see 3 main branches here in asterisks, and that this is the minority branch here before infusion, and that by 1 month post-infusion, we see no viruses on this branch. We look genetically at these sequences. Here I'm just showing the conservation of the CD4 binding site. So here, loop D. This is if you take 1700 subtype B sequence from the Lannel database and look at how conserved all these residues are. You can see they're pretty much, the sequence D or N and A is almost 100% conserved. What we see in this person pre-infusion is that all the sequences here have an unusual lysine at 281, which is almost not ever seen in the Lannel database. Here, what we see when we I'm taking just the cloned envelopes and generating a phylogenetic tree that's color-coded according to phenotype. So here we can see that this top branch is the most sensitive to VRC01, and this is the branch that has the canonical DNA sequence in loop D. On this branch, the pre-infusion sequences contain this unusual leucine, and they're pretty resistant to VRC01. But post-infusion, what we see is in black, sequences are all on this branch, but are completely resistant to VRC01. And when we look within the epitope, what we see is a selection for a very, very unusual serine at position 280 that, again, is almost unseen in the Lannel database. And here, we have this unusual loop D and no change in V5 sequence. But then again, by 2 months, once the antibody leaves, we see the wild-type sequence branch come back. And then just in these 2 people who remained suppressed, what we did was take sequences from 60 days when virus rebounded, when antibody was gone, and we looked phylogenetically and phenotypically if we could detect selection by VRC01, and we don't see any. So you can see here, all the sequences in green and white are totally commingled. We can clone and see no difference phenotypically in resistance to VRC01. So essentially, there's no selection that we can detect by the antibody, and the same thing with this person. So based on this data, I had hypothesized that plasma virus load means something, and that when you start off with low plasma virus load and put in a monoclonal antibody, that then you can see possibly full suppression, and that's what we saw in the VRC01 trial. So I actually thought that in these trials where they did analytic treatment interruption, when you're starting from full suppression on antiretrovirals and put in a monoclonal antibody, that these people would be able to maintain suppression, and that is absolutely not what has recently was seen in these 2 trials. So and I'll just briefly go through these, where again, there were 3 trials, 2 with VRC01, so these were put into one paper together, and this is the one that I'll talk about because I did some analysis, but there's also this paper using 3BNC117 from the Rockefeller group. And so overall, what you see in both trials is there's a slight delay in rebound virus when you take people off antiretrovirals in the presence of monoclonal antibody, but you see virus rebound. You see genetic restriction and selection for increased resistance, again, in everyone that was looked at. However, there were some big differences in that the 3BNC trial pre-screened people for sensitivity to the antibody, and they saw a much longer delay in virus rebound and much more genetic restriction. But I'll briefly talk about the VRC01 trial because these are the ones I analyzed. So this is 2 trials from ACTG and the NIH. both using VRC01, and then here just the idea is that these people are have suppressed virus loads on ART in blue, and then they are infused with VRC01 here in green. Then they are taken off ART here in red and monitored for virus load and given multiple infusions of VRC01 about every 2 weeks until the virus begins to rebound, and then they are put back on antiretroviral therapy. And so here's just a summary of the 2 trials, and you can see there is some delay in virus rebound, but really what's striking to me is that there's very few, you really see delay in just a few people in both these trials, say 2 people. And then here's a summary of the data compared to historical controls from ACGG trials. In blue, time to rebound, the NIH trials in orange, and the ACTG trials in green. And so the summary is that at 4 weeks after treatment interruption, there's a statistically significant difference between monoclonal therapy and not, but by 8 weeks it disappears. And so the ACTG trial cloned plasma virus from these people, and how they were able to do this was they had time points before these people went on antiretroviral treatment. So pre-ART sequences are in black, and then post-infusion virus rebound sequences are in orange and red. And what was striking about their findings is that they see, in a couple people, especially this person, really monophyletic rebound. And so Tim Schacker showed yesterday, in the absence of monoclonal therapy, that most of the time you do not see single foci rebound. You see multifocal rebound after treatment interruption. And this was seen in a couple people, but it was very striking that you do see some genetic selection here. And then when you clone these envelopes, they did see that these the rebound viruses were more resistant to VRC01. And so I collaborated with Taiwook for the NIH trial, and we did things slightly differently, where he actually looked at replication-competent virus isolates from pre- and post-infusion. So he just took PBMC and did virus outgrowth assays, and then looked at sensitivity of outgrowth virus from the reservoir directly pre- and post-infusion. And again, he saw, and so it's graphed a little differently. This is suppression of HIV, and so less suppression lower down means increased resistance to VRC01, and he saw in almost every case that virus became more resistant to VRC01 after infusion. And then here, I just wanted to highlight, so we took plasma genes similar to the ATTCG trial, where we had samples from the NIH before these people went on antiretroviral therapy, and then compared it to the plasma rebound virus after infusion. And so here I'm just showing maximum likelihood trees of the genes, and you can see that all these subjects are subtype B infected, and that almost every single person has a diverse quasi-species consistent with being chronically infected, except this person who I suspect went on antiretrovirals during acute therapy, but we can't confirm that. And so here, I'm breaking it down individually and showing before and after infusion in red and blue. And so pre-ART is the blue circles, and post-VRC01 infusion rebound is in the red circles. And so one thing that's striking to me is we do not see in the NIH trial the sort of monophyletic low diversity that was really striking in the Rockefeller trial and seen in 3 out of 12, I think, subjects or participants in the ACTG trial. What I can say is that N2 and N3, there is lower diversity in the rebound virus as opposed to the pre-ART virus. And so you can see that there's some genetic restriction here in this person. And this person didn't have diversity to start with. But for the most part, you can see, especially in this person, a lot of virus diversity in the rebound virus that is related to the pre-ART virus. Oops. And so we started cloning some of these plasma genes from the NIH trial. And what I want to point out is that these people are starting off slightly more resistant, I think, to some other people. And so I'm trying to compare this to the previous slide I showed. So this is the viremic subjects who were infused with VRC01, and this is the suppressed subjects who were infused with VRC01 at the NIH clinic. And the scales are slightly different. But if you look around 1, these are IC80s that are graphed. kind of looks to me like in general, these subjects are slightly more sensitive and have lower IC80s than these subjects or participants here. And so I think moving forward, there's a question of, is 3-BNC117 more potent than VRC01, and that really makes a difference clinically when virus is rebounding? Or is there something about suppression and rebound that these viruses tend to be more resistant to VRC01 than, say, just taking envelopes from chronic infection? And so here I'll have a couple conclusions from these studies, that a single antibody, and again, here I'm taking into account all the studies that I've shown you, not just the ones that we conducted at the NIH, a single antibody infusion can decrease virus load in the plasma and exert selection pressure on the virus quasi-species. You can see that it's exerting some effect. Each individual's quasi-species during chronic infection display a range of sensitivities that are diverse within each person. They display a range of sensitivities to broadly neutralizing antibodies. However, in almost every person, you can see evidence of selection by the antibody. Virus populations that become more resistant to the antibodies also maintain resistance to autologous antibodies. So you see existing immune pressure from the person in addition to selection pressure from the infused antibody. And for the most part, virus that is sensitive to the antibody does return when the antibody leaves. But what I really want to say is that more than any conclusions from these trials, I think there are just so many more questions that remain unanswered about monoclonal therapy. And so I tried to think of all the main questions that I still have. What is the relationship between these in vitro studies when we talk about broadly neutralizing antibodies, and then what happens clinically? Clearly there's some relationship because you can see people who are fairly resistant have less clinical effect. But are there more subtle differences that we haven't yet explored? Are there the potency differences that we see in vitro between VRC01 and 3-BNC117 explaining some of the differences that we see clinically in vivo? Or is the difference that only some neutralizing antibodies target infected cells, and so we see more effect in some cases than others because the antibody is exerting more functions? Really, all we're measuring in the in vitro assays I talked about today is neutralization, which, as everyone has mentioned, is only one function of an antibody. And so the ability of antibodies to mediate ADCC or ADCIP, et cetera, I think can be broken down into many different parts. And basically, neutralization, the antibodies are binding to viruses. To opsonize infected cells, they need to be able to bind to the conformation of envelope that's on infected cells. And that is just beginning to be explored by different groups. And so the best neutralizing antibodies may not be the best antibodies to use for in vivo clinical trials where you really want to target infected cells as well. Also, where and when is the antibody being distributed? In the participants who have ongoing plasma virus replication who are not on ART, you can infuse antibodies IV and you see a drop in plasma virus load. Looking in the plasma may not be where we need to be looking in trials where people are already suppressed and have no plasma virus load. And so in these analytical treatment interruption trials, when you are giving the antibody for 3 days and then taking them on antiretrovirals, what tissues is the antibody going to? We know that antibodies can be distributed to all the different tissues in the body, and we know that antibodies can go to the lymph node. But when are they going? And so timing should also be explored. And specifically for these broadly neutralizing antibodies, prove that they're going to the lymph node and gut, even though I suspect that they are. Other questions that I have are, is it easier to escape from some antibodies than others? 101074 seemed pretty easy for the virus to escape from it. One question is it just because it's easy to see and it's harder to see because it is a continuous epitope versus a discontinuous epitope for CD4 binding site antibodies? Or actually, is it pretty easy to escape from these V3 glycan antibodies? And I suspect Ron might have some data about combination therapy, and so I think that would be really interesting, what are the best combinations of antibodies to use to prevent virus escape. And so I'd like to acknowledge everyone. All these studies were done sort of as I finished my postdoc and transitioned to be a professor at George Washington University, but all the clinical samples were from NIH trials at the VRC and Tyvek's trial at the Laboratory of Immunoregulation. So I'd like to thank everyone, especially my mentor, John Mascola, and all the study participants. Thank you.